Good afternoon, everyone. I think that we're quite a few people now, and I would like to start. I would like to welcome you to our first webinar on the SOP development that was prepared by Rebecka and Anna. But first of all, I would like to introduce myself. I'm Gina Kardik from the European Forum of Farm Animal Breeders. And what we do is we represent breeders in Europe for different species, poultry, ruminant, pigs, aquatics, and also insects. The aim of EFAB is to represent the members' interests at the EU level, both for policy and legislation, but also to promote responsible and balanced breeding. Our role is to also engage in dialogue about animal breeding and reproduction, but also to act as a knowledge provider, so that is translating sciences for the wider society. And this is what we've been doing for different projects, but we're also part of European Livestock Voices. And with Code FABAR, it is the Code of Good Practices for Responsible and Balanced Breeding for different species. And recently we have a Code of Good Practices for insects too. But we also are a sister company for FabreTP, which is a technological platform. So what it covers is also knowledge centers and academia, so different universities. And together with the breeders, we try to bridge the gap, but also promote and support research and innovation in animal sciences. And we help to develop the research and innovation agendas to set priorities for upcoming years. And most of all, it is important in the sector that we're working on to bridge the gap between the private and research sectors so that we can provide better understanding of animal breeding and genetics. I will not take more of your time. I would like to introduce Rebecka. Rebecka is the one who's going to do the webinar today. Would you like to say something about yourself, Rebecka?
Yes, hello everyone. My name is Rebecka Westin. I'm a researcher at the Swedish University of Agricultural Sciences, and I am also the VP leader for Work Package 2 in the Pigweb project. So that's why I'm here today. And with me, I have my colleague, Anna Wallenbeck, who will also present for you today. She's also a researcher at the same department as me at SLU in Sweden. Okay.
So I will let Anna, share her screen.
Should I share, Rebecka, or will you share?
I can share. Okay, do you see the right screen now? Perfect. Okay, I won't see you now if you raise hands, but we will have a lot of time in the end for questions. But I'm sure if you have questions, someone else will see that you raise your hand and tell me if there are any questions along the way, and that's fine. So I want to start in this end, talk a little bit about what is quality in research. Quality in research can be a lot of things, of course. And first of all, it has to be a good match between the methods we use and the research questions we have. So we get the right answers on the questions that we have. And that can be about how we select our subjects or the sample size. It can be about that we do correct measurements of the outcomes. We have to avoid the systematic or non-systematic bias. And of course, we have to think about our hypothesis and use correct statistical analysis. And appropriate conclusion given type and quality of data registration and the analysis. So there is a lot of things we have to think about. But I think some of you at least are attached with the research farms. And when it comes to research farms, it can be a bit different. Overall, it is about to provide the necessary information to reproduce or be able to replicate the study when we inform other people about the study. So when we come to our research farms, what is then quality? And then I think it's again about the study planning and design that we do. This design of the study, is it an experimental or observational study? But when it comes to the farms, it also is important that if it's a long-running experiment, all pigs should be cared for in the same way during the whole trial, even if we have a change of staff, for instance, which is common. It's not the same person taking care of the pigs every day. And as a researcher, if there happen things on the farm, I would like to know that. So it's important that we have a good documentation on deviations, I think, on the farms, both if it's a research farm or if we are out in the real world. We use a lot of different equipment in the research farms. We have scales, and there are different equipment that stands. It is not used every day, maybe, but when they are used, we should want them to be correctly calibrated and taken care of. And that is also something that I, as a researcher, really want. So, and do I know that that is taken care of? And all of this to get reliable data, of course, and that is comparable over time. So again, the farm has to be able to provide the necessary information that we need, so we know what is done and we can reproduce it in the future if we want. And do we even need research farms? Why should we have research farms? I think they can provide a more controlled environment compared to if we do research out on a general farm. And they can do more standardized basic management, hopefully at least. And most of the research farms, they have more staff working there, so they can do more detailed and more extensive recordings than you can do on a out in the field. But it's also important to remember that we should do both, I think. Some studies are relevant for research farms, other studies maybe it's better to do out in the real farms when there are more variation. And it's important still that we have the research farms. I don't know if you recognize the man on the picture here, but that's actually the Swedish king and his son, Prince Carl Philip, who was visiting the Swedish research farm that we have in Uppsala, Lövsta research farm, where we have pigs. But there are some challenges with these farms because they are quite expensive to run. But they also share some common challenges. And every farm, they do some basic pig management, and very often more or less the same standard traits are recorded, like weighing the pigs, for instance. And that provides possibilities to have standard operating procedures that could be shared across facilities. But researchers are not always aware of that there are SOPs and their impact on the quality of their studies and data. And also facility staff are not always aware or used to working with SOPs. And maybe they are not aware of the potential impact a deviation from the standard procedures can have if something is done differently. So we think that to safeguard good research and data quality, this has to be more, we have to be more aware, and we should try to improve this in our different research facilities. So that's where we come in then. That's where Pigweb comes in, when we do have this task where we have a goal to harmonize protocols for basic management and recording of standard traits. in experimental research facilities. So that's the aim we are heading for. But just as a start of what is actually a standard operating procedure, I'm not sure you all are familiar with that term. And very simple, it is an instruction to follow when performing a specific procedure. And coming from Sweden, I think the most excellent example is our IKEA instructions. I think you all have seen them. That should be really simple, a lot of pictures, not a lot of text, and anyone should be able to follow them, which mostly is true. I think sometimes you wonder what they mean anyway. But so think of that as that is a standard operating procedure, like the instruction book for how to do things, and that we can apply in our farms, and they can look however you want in a way, but you need some general information. What is it? What instruction is this for? And you have to describe the procedure. You have to make a list of the equipment you need. If you need to calibrate it, the description of how calibration works should be there as well. And then you should have a description of what type of registrations you should do and how to document them. If there is a special form for or what you should fill in when you do this procedure. And why should we have them? Apart from what I've said about the data quality, I think it really, if you have written instructions, then you have something to discuss with the staff at the research facility, and with discussion, and then you can get harmonization in the management and recording among the staff. But if you don't have anything like to start from, then it's very likely that people will not do the same. There will be quite a lot of differences in how people do things. So written instructions help to get this harmonization between the staff. It also facilitates communication between staff and researchers. If I, as a researcher, come out to the farm and I wonder, how do you do, what feed do you give or something? It could be about anything. And if they can tell me, but if I have a paper with written instructions for this procedure I wonder about, then it's really easy for me to bring that home and just write it right into the material and method section if I write a paper, for instance. So it really helps in the communication. And if I want to change anything, that could be as well as a researcher. Maybe I want something to be done different. That's also easy. If I know exactly what they're doing, then we can discuss, okay, you were doing this, but now I want it like this. So you have a good start for your discussion then. And if you have a clear instruction, it helps you not to forget things. If you have a list of exactly the things you need, then you hopefully don't forget what you need and have to go and get it, or so there's not a short supply of the materials you need, for instance. So that is very helpful, and especially if you have new employees as well, like that it's really clear for them what you need as well. And if you have SOPs, absolutely you should revise them, and that helps you with to constantly develop and improve the management on the research facility. So there's a lot of good things here. And when we did a talk with our staff manager at the Swedish research farm, they have done this journey. They have developed already before we started this work in Pigweb. They had made a lot of SOPs. And Elin, who is the manager, she said that, yeah, it takes time to develop these SOPs, but once you have them, they save you a lot of time. So she was really happy that they had done this. It was quite felt like a big challenge in the beginning, but now when they are there, all are very happy that they have all these written instructions, and it saves a lot of time. Especially in, she said, like when you have new staff, it's super easy to get them to know how we work on the farm and so on. This is one example. This is from Wageningen. They are really good in this. They have been working with SOPs for a long time. And this is an instruction for earmarking, for instance, where you have first some general information that is in Dutch. Sorry for not translating all of it. But then you can see like the purpose of this SOP, and then the list of materials that you need. And then in this case, they have a section for safety precautions. And before you start, what do you have to prepare? And then the instructions, how do we perform this procedure? And then some references. And in this case, they don't have any documentation that is applicable. So it's 2 pages of paper in very simple dots. That's one way how it can look like. Okay, now I will hand over to Anna.
Yeah, I will tell you a little bit about what we did in this Pigweb Task 2.1, where our aim was to improve and harmonize protocols for the standard management and recording in pig research facilities. So what we did in 3 steps. We started to identify the key areas of standard management and recording. And we'll come more into that. It's what are the standard management and what are the standard recordings? Well, then it's not the things that we do specifically in trials within feeding or within behavior or within whatever area. It's what we do with the pigs all the time, regardless of the subject of the trial. We started to identify what areas this was. Then we developed improved protocols based on the current practices in the different research facilities that were included among the partners in Pigweb. And then we did a SWOT analysis on how to implement and use these protocols in the same facilities. You can move to the next slide. So in the identification of these key areas, when you see the results, it's kind of, yeah, of course. But we had long, very interesting workshops and talks among the people. We were quite many partners and quite many people working with pig research. And it took us some time to do this identification. What we did was we started with an inventory of the current standard management and the traits that we record and used in the different pig facilities. And we did that with a rather meta questionnaire, like we did an overview questionnaire. We didn't steer it so much. It was more to get the brainstorming or getting the different, what's done, an overall inventory, not too detailed, more overall. In the next step, we developed definitions for the standard management and recordings. And we did this because we needed those definitions. We also needed criteria for what are the criteria for management and recording to be standard and not being specific for specific areas. And we did that in a workshop. And after that, when we had found the key areas, then we went back out to the research facilities and made a more detailed questionnaires. Okay, so within these key areas, what are the current practices? What are the current SOPs used? So also the first step we did in 3 steps, and it was quite a process and a very interesting process. Next step, the next slide. So these were actually the key areas that we identified and that we continued to work with. And so it was stable climate, enrichment, feed, sow body condition, piglet mortality, pig weight and growth, pig health, and abattoir, so standardized documentation on how things were handled at the abattoir. Next slide. So the next step then was that based within each of these key areas, we developed an improved protocol based on the current practices used. And here you see one of the drafts, but if you you will get this presentation later. In the QR code, you can find the protocols published on the Pigweb website. This was also done in steps, I must say. We had quite a lot of steps before we had the protocols done. In the next step, all the different partners took these protocols, went back again to the research facilities and made a SWOT analysis. So if we should use for each of these key areas, we had a protocol. And then we went through them in detail together with the staff. In some cases, it was also tested practically, but in general, we wanted to discuss how would it work to implement this SOP or these procedures in this facility. So for each of the protocols, we had strengths, weaknesses, opportunities, and threats for each protocol from each of the key area. So this was done in all the different partner organizations and countries. We can move to the next slide. So gathering all this information, having this process of development, and then this process of developing the protocols and assessing the protocol. What did we find? Well, the procedures varied very much between facilities. And coming back to what Rebecka just talked about, the precision and what's needed, what's quality in research, it doesn't have to be bad that it varies, but then we need to be very aware that it varies. It was also that many facilities, even though they had adequate routines and did things in an adequate way, they didn't have any SOPs where this was documented. Actually, only 2 facilities had written SOPs for more than one. Some had for a few of these key areas, but only 2 facilities had for a number of these key areas. And the key areas with potential of improvement and harmonization that could be seen as these basic things that you need regardless of what your research is about. You need to have this information standardized, and it's very important in the communication with the researchers how this is done. And those are the stable climate enrichment feed, cell body condition, piglet mortality, pig weight and growth, pig health, and what's done before and at the abattoir. Then I think I hand over back to you.
And we can say that if you go and look on these protocols now or after this talk, you can see that they are mostly about documentation. Actually, they are not so much SOPs with descriptions of how you should do the procedures, because we realized on the way that we do so differently. So it's a better idea to start with like how procedures are done, to do SOPs like based on your own facility, than trying to make one that looks the same for everyone, because there are so different like in how the stables look and so on. So these protocols are more about how to document that you have done things in a way. So when you see them, you will see that they are not so SOP like in a way. They are more about what standardized documentation. Yeah. So, but that in a way, when we did this, those protocols were in a way the end of our task in Pigweb, but we think that it needs more than only having a few documents. So that's why we're having this webinar now, because we we feel that a lot of the research facilities, when talking with them, they were like in the start of wanting to doing this. So we will share some of our tips and tricks that we got from the discussions with these different facilities. So we can help, I hope it can help you a bit on the way to start making your own SOPs at your own facilities. So the first tip we have, or that people said to us, was that don't set up a plan to make all SOPs at once. Then it will be too much and you won't get them, you won't cross the finishing line. So start with one, make one, and then gradually you increase the number of SOPs. So start with a procedure that you do often, and then you make that one until you have done that. And as soon as you have the first one is the most difficult one, but when you have it and when you did the first, then it will get easier and easier. So start with one. Don't think that you should do 20. Start with one. And to help you with this, we have done an empty template that you can use. And of course, the other protocols you have for inspiration as well. But if you go to the Pigweb website, you will find this. So we have made a template. It's in Word, so it's just to download and start writing your own text. You have some examples in gray here, but that you should, of course, change so it fits your facility and what you want to add to it. But I think this is a good starting point. Then you don't have to, you don't have to invent the wheel. You have the wheel here, so you can start and build from this. And just focus on describing the procedures as they are performed right now in your facility. Don't think about harmonizing it with other facilities, or just write down how you are doing it right now. And don't worry about it being too general or too broad. Just to have something on paper will feel very satisfactory in the start. And then you will go through revising processes, and then you will make it better and better. So, but in the beginning, it doesn't have to be perfect. So just focus on describing what you are doing, how you are doing things. And you should think that they are like a key in your continuous improvement work. So think about the strategy for revision. At SLU here in Sweden, they revise these twice a year. And then they read it through and they think, yeah, do we still do it like this? Or have we changed anything? Or is it the best way of doing it? Should we think about something else? So that it doesn't take a lot of time, but it's good. They discuss it among the staff together. So it's a good way of reminding each other, yeah, this is how we do things. So it's not a big thing to do the revision, but it's important to have it and to plan for it, so it will be done.
I can fill in a little bit there. So it's a good tool for the communication around the management in general. And as this is, I mean, you can have SOPs on anything, on how to take blood samples, but that's not the basic management. In this case, it's also together with the basic management of the entire herd. And with this last dot here, connecting back to the first one, don't make all SOPs at once. The important thing is to start with a few, the most important things that you do every day, and that it's this gradual improvement, and the continuous improvement work is part of it. So that in that you can also include adding on new SOPs along the way. That's just what I wanted to connect those 2 points with each other.
Yeah, exactly. And I think, you can also think not to make them too detailed, because then you have to revise them very often. I don't think that is a big problem in a way. If you write down what you do, then you have details enough. But this is the last point is really important, and it's about this. Make a habit on using them for internal communication among the staff, and especially if you are handing over working tasks or introducing new staff, then you have a really good example of when to use them, but also for everyday use in a way. And then in your discussions between staff and researchers, researchers should be aware that you now have your SOPs, and it helps you a lot, I think, in discussion between staff and researchers. So it's really a very good tool, and that it should be used. It's not that you do these and you write these instructions and then you never look at them again. They should be looked at and used when you communicate with others. This is just another example of how an SOP can look. Again, we say very much thank you to Wageningen providing us with these. So here you have one for weighing of finishing pigs. And we have the template you will see is very much based on this kind of SOP. So I think this is really something that everyone can do. And you can add pictures as well, for instance. If you go to the Pigweb website, you will find the abstracts, and you will also find that we did a practice abstract with these points that I have showed you now, a little bit of background and what is an SOP. So if you are not yourself working in the research facilities, I'm happy that if you can share this with your staff managers and so on, that would be great, because we really hope that more of you will start developing your own SOPs. And I think maybe, yeah, this is how it looks again, and you will find not the practice abstract is only in English, but all the protocols we developed, they are available in 8 different languages in addition to English. So you will that also helps with communicating with the staff, of course, because English is maybe not always the used languages on your farms, or it isn't. So that will hopefully be helpful as well. Some conclusions. After doing this work, we can conclude that procedures varies between facilities, but there are a lot of key areas where we do the same things, and that we can see that there can be improvements and harmonization, at least within the facility between the staff, but also to some extent maybe between facilities. But I think the most important is that it should be harmonized within the facility. So your staff do all staff do the same. And we can conclude that when asking around then for what facilities have SOPs already, very few did actually. So there is a lot of potential here to do this work and start to use SOPs. And if you don't have them, start small, develop it on current procedures, add on gradually, and this will lead to harmonization within your facility. And now you have a template and other protocols, so use them for inspiration when you start developing your own. And we suggest to revise SOPs twice a year. And when they are in place, they will save you time. So even if you think it's a lot of work to get them done the first ones, they will save you time later. I think that was my last slide. So thank you very much. And we are very happy to take on any questions if there are any.
Thank you, Rebecka. If you have questions and you are shy, you can always put it in as a question in the box and I would read it out loud, or you can raise your hand and I will give you the floor. No questions?
I guess you will send out this presentation.
Yes, I will. I will send it to them and then you can the participant can always come back and ask questions. But maybe I can ask you, since you've been on the field, what do you think are the main challenges in building up, in coming with that template at the end of the day, you know, on the research facilities?
I think the challenge is that it's something new and that you're not used to work with these documents. So, and maybe, yeah, since they haven't used them, they maybe don't know why should we have them. So I think it's and I think I most heavily experienced from the Swedish research farm, and they were like in the same in the beginning, like, yeah, but why do we really need them? But now when they have them, they are really happy that they have them. So it's something they really didn't regret, although it took some time in the start to make them. But and that was something that come from a top. It was not something they had asked for themselves. Yeah. It was they had to do it in a kind of help me on a certification.
Yeah, in a ISO certification system for both quality and environmental aspects. And I actually worked in that herd at the time, and there we had to do everything at once. And when you have never done it, you get the picture that it's so complicated. And now in this case, we looked at the basic management. In that case, we needed to do it of everything. Also, like when we do more invasive things with the pigs. And that is more logic. But doing these basic things felt a bit, but we've always done it like this. And it was a lot of work. And that's I think one of the things that could be learned from there. And I know that it was mentioned in other in facilities in other countries as well, is that don't do everything at once. And once you have done it and you start having this update. Also, when we had to go through at the Swedish herd with the staff that has worked with this for a couple of years, they were, oh, but this one is not now, but we have changed this. We need to update this one and we need to do that. And yeah, we have a much smarter way of doing this now, and documenting that is so important. Of course, we do it when we have a trial and a researcher come there and have the look on you that I need all this information. But if you do it all the time, afterwards, you like when we did that revision, when we sat there, oh, we need to revise this one. It was something on castration, I think. So it was not even within what we were doing. But and it was like, yeah, because this is so good. Then we have this and we have documented it, and all researchers can see it at once. So everybody who's there doing the research, they get the information and it's updated at once. So it's a very good way of communicating within the herd, but also externally with both the researchers and students and whatever.
Yeah, no, that's interesting. So I hear from you then that the SOP, the challengers, is to at first to make people accept it, but then on the long term, the result is that it helps them in management and farming audit on farm that kind of make them their life completely easier. And yeah, I mean, it's interesting because many times it's so SOPs are so simple that anyone can make it in a certain way with that thing, where you see that sometimes in accessibility to farmers on how can they provide information on what they're doing is a big problem in many farms. And I think SOPs then can therefore help in providing that accessibility and then easy up their life with what is things that for them are not important in their head, you know. No, I think it's super interesting. And if I would definitely share the PowerPoint, and the webinar would be available on YouTube for everyone. So we'll keep sharing it, and I hope that if you have questions in the audience, you can always come back to us, to Rebecka, Anna, or me, and I will forward it to both of them. Rebecka and Anna, I really thank you for your time and appreciate the work you've done on the SOPs and the practice abstract. And I think it's great work, and I promise that I will try to disseminate the practice abstract in different languages so it's more accessible. If anyone would like to see the practice abstract, it's on the website, and the QR code will be available on the PowerPoint presentation of Rebecka and Anna. I thank you for your time, and I think we can close the meeting today.